Hi Rav, okay, so you've said that your textbook, when looking at the biochemical tests for substances, and you've cited the Benedict's test there, it says that a more accurate way of doing that is to either basically take the precipitate that is formed. When we do the Benedict's tests, we take a liquid, and we get a solid precipitating out. So that amount of that solid, the more solid there is, the more sugar there was in that solution. And we can therefore weigh that solid, and then we get a quantitative reading. So actually what the spec says is qualitative methods for determining the biochemical test. So we've got Benedict's tests, we've got the emulsion test for lipids, and we have the Biuret test for proteins. So these are quantitative measures. OCR will ask these things, potentially. AQA, it's possible. It would be much more likely to come up as an assessment objective 3, a suggest. So like pushing you a bit beyond the specification. But I'll go through them with you. So yeah, essentially the amount of precipitate formed, which is a solid, is proportional to the amount of sugar. We can therefore weigh it and get a numerical number for how much precipitate there was, which is directly proportional to the amount of sugar that was in the solution. The other method that you've mentioned is to use a colorimeter. Again, we're going to get a numerical, a quantitative answer. So the color change that we're observing with the Benedict's test, the qualitative Benedict's test we can measure that color change with a colorimeter. And therefore, we are going to be able to get a number value, which is not a qualitative or sugar present or not present, or a broad spectrum of color turning from the blue negative test to the brick red positive test. You can obviously end up with it anywhere on that color spectrum in between, and you could look at it and say, it's roughly halfway between this and that. But with a colorimeter, you would set up a calibration curve in much the same way that, again, all my lessons in the course will cover calibration curves. So you create a calibration curve with known concentrations of sugar. You'd then perform the Benedict's test with them, with the same volume of Benedict's reagent, all that kind of stuff. You'd then take those readings on the colorimeter and produce the calibration curve. You'd then test your mystery substance with unknown concentration of sugar. You'd do the same, keep all your variables exactly the same quantity of Benedict's reagent. You would then measure on the colorimeter what the absorbance or the transmission, depending on how you've set up your colorimeter. You'd get a numerical number, which you can then read across on your graph to then figure out how much sugar was in there in a quantitative way. So essentially, there are two different methods that you can do that, but they are both quantitative methods and not qualitative methods. And I can tell you from experience that AQA are not very likely to ask that kind of question. And again, if you check out the topic revision pack for the carbohydrates and lipids topic, I have separated the biochemical test there. So you can do all of the questions that AQA have ever asked on the biochemical tests pretty quickly, because there's not that many of them. And then you can get really comfortable with what that feels like in a real AQA biology exam. So good luck. And if you have any follow-up questions on that, you know where to reach me inside the TT Academy. I'm there.